Isolating DNA from plant tissues can be challenging as the biochemistry between divergent plant species can be extremely different. Unlike animal tissues where the same tissue type from different species usually has similar characteristics. CTAB is also called hexacetyl trimethylammonium bromide is a cationic detergent that facilitates the separation of polysaccharides during purification while additives, such as polyvinyl pyrrolidone, aid in inactivating polyphenol. CTAB-based extraction buffers are widely used when purifying DNA from plant tissues. To overcome the challenges presented by plant tissues, this method has become the go-to protocol for DNA extraction and purification from leaves and seeds. It was developed in the 1980s and has been used ever since.
How to extract plant DNA by CTAB method. CTAB is cetyl dimethylammonium bromide, and this technique was developed by Doyle and Doyle in 1987. It's a very widely used technique for DNA extraction, and DNA extraction is one of the most important steps in plant biotechnology. We require a plant tissue, and we are using cauliflower. We are using cauliflower as the source of plant material, and we require three important reagents. One is the CTAB. This is the main buffer, CTAB buffer. We have prepared and kept it. This has to be kept in the water bath because it has to be heated up to 60 degrees centigrade. You keep it in the water bath and keep it there. There is a glass slide there, and then we require this solution. It's a mixture of three chemicals. This is phenol, chloroform, isoamyl alcohol in 25:24:1 ratio. This is the other chemical we require, other reagent we require. And we also require isopropanol. So three important buffers are required. First step is we have to weigh 200 milligrams of cauliflower, and then you have to homogenize the plant tissue in 1 ml of CTAB buffer, and the penicillin water should be kept in the fridge for at least half an hour because you are using the cold penicillin water, it should not be hot. Then the homogenized plant tissue is transferred to an F&R tube completely. So whatever you homogenize, transfer it completely into an F&R tube, and then keep the tubes in the water bath, which is preheated at 60 degrees centigrade for 30 minutes. Then centrifuge the tubes at 12,000 rpm for 5 minutes. Transfer the supernatant to a clean F&R tube, second tube, and then add equal volumes of phenol, chloroform, isoamyl alcohol in 25:24:1 ratio. In the F&R tube, you have to centrifuge second time, again at 12,000 rpm for 1 minute, and you get the three layers in the F&R tube. The bottom layer is the organic phase, which will contain the lipids. The interface layer will have the denatured protein, and the top layer is an aqueous phase, and we need this aqueous phase. The DNA is there in the aqueous phase. Carefully transfer only the aqueous layer into one more F&R tube and add equal volume of isopropanol. Once it is there in isopropanol, you have to incubate at -20 degrees centigrade for 20 to 30 minutes. Then the threads of DNA is precipitated at the bottom. You have the supernatant where the DNA is not there. DNA is there at the bottom. Then you have to remove the supernatant and throw the supernatant. Only take the DNA as a pellet and dry it. And you have to wash this with 75% alcohol 3 times. So put some amount of 75% alcohol and then centrifuge for 1 minute. Then take out the supernatant, only retain the pellet, do it 3 times, and finally you get the DNA threads, and that DNA you can store it at -20 degrees centigrade, and later you can use it for either cloning. You can also run it later on a gel to study the DNA. So for all further studies, this DNA that is extracted, which is stored at -20 degrees centigrade, can be used. So this is what is Doyle and Doyle method of DNA extraction from plant material. And this is a very popular technique and widely used technique in plant biotechnology. This is DNA, and we have it in isopropanol. It should be stored in -20 for 20 to 30 minutes. Later, it can be washed with 75% alcohol 3 times to get a pure form of DNA. Otherwise, we can store it in -20 and use it later. And whenever we are using, you can further wash it with alcohol and use it.
Thank you for watching my videos. Please like, share and subscribe. We'll meet again in the next video with some other topic. Thank you very much.